Hope everyone is nice and safe out there in these challenging times. I just wanted to extend a warm welcome to everyone to the webinar. As Charlotte mentioned, my name is Matt Davis from Millar. I'm the Director of International Sales and Business Development and I'm based in the UK. So I work closely with ADInstruments in a global capacity to help support all our existing and new clients to make sure they have the best user experience with our technologies. So in my session today, I'll be discussing accurate pressure recordings in non-cardiovascular applications. So I'd also like to start off by thanking Dr. Xu for his talk today. I'm always amazed by how much he can get out of a pressure signal, the parameters he measures. He's been a long time Millar adopter, and yeah, really nice to see his research there. So for all of those who are attending who are new to Millar technology, I just wanted to begin with a short overview video to introduce Millar as a company and what we do.
Since 1969, Millar has been a pioneer in the development of advanced pressure sensor technology to fuel groundbreaking research. Around the world, animal researchers rely on Millar technology, including pressure catheters and PV loop systems to make measurements and decisions with accuracy and confidence. Millar's clinical product line proves we understand what it takes to bring a commercially approved clinical product to market. Delivering precise measurements in real time, Millar products are routinely used for benchtop testing and clinical trials, often serving as a bridge to OEM medical device innovation. By partnering with us, you can leverage over 50 years of MEMS manufacturing expertise, backed by thousands of publications across hundreds of applications. As the recognized gold standard for accurate pressure measurement, Millar technology has been instrumental in medical device validation and pharmaceutical discovery, and is used routinely in advanced medical applications. Working with an established medical device company with proven technology also reduces the risk associated with integrating MEMS technology into existing product development plans. Millar has a proven track record of delivering low drift. Highly skilled OEM engineers follow the collaborative processes we've developed over the decades to integrate our market-leading pressure sensors into fully functioning devices with the lowest drift on the market, enabling innovative discoveries for life science and clinical researchers around the world. Millar is committed to bringing together proven technologies and breakthrough ideas in order to impact medicine and make the improbable possible.
So I hope the video provided a small insight into Millar and our business. Essentially, we are a pressure sensor integration company, and we have 3 business units. So we have OEM is part of our business. That's where we integrate pressure sensors into mainly medical devices. We have a clinical arm to our business, which is where we supply clinical diagnostic pressure catheters into the cardiovascular, airways and compartment pressure markets in Europe and the USA. And then we have finally the life sciences or the preclinical market, which is our focus for today. So the topic of today's webinar is accurate pressure recording in non-cardiovascular applications. So the choice of this topic stems from Millar being widely recognized for pressure and pressure volume measurements in the life sciences in the cardiovascular area. So we wanted to discuss topics outside of this well-known application and showcase other applications for Millar pressure technology. So just a quick dive into what we want to discuss today. So firstly, we just wanted to look into some of the challenges in designing and manufacturing sensor-based pressure catheters. We'll take a small insight into drift, and then we'll move on to some applications insights. So we're looking at intracranial pressure, IFP or fluid, interstitial fluid pressure, and airways pressures. And we'll do a small look at sleep apnea as well. So Millar and ADInstruments have been partners for some 20 years, with the last 5 or 6 years where we've been in an exclusive partnership in the preclinical research field. So the core value for our partnership was based around providing seamless solutions for researchers that would allow them to produce highly accurate data that they could trust. We're also conscious that research funding across the world is challenging to obtain, so we want to provide the best-in-class solutions to all our clients for them to be successful. The statement on the screen resonates with everyone at Millar and ADInstruments in that a research study is validated by its accuracy, and accuracy begins with the selection of instrumentation. So sensor integration. A lot of people think that those signals are really easy to get, but the manufacturing behind a Millar catheter is quite unique. So sensor integration has been around for many decades. Millar was one of the pioneers of pressure sensor integration, having placed sensors into catheters in the late 1970s and early 1980s for human use applications. So all the catheters we produce are handmade. The reason many of our processes are still done by hand is because we work literally at the limits of what is possible at this scale. So as an example, we still have been unable to find a vendor capable of automating our wire stripping or epoxy deposition process. We have been looking for someone to do this for us for over a decade, but have yet to find someone. So there's sort of a three-part process. It all starts with the sensor. So we utilize MEMS technology, which is micro electromechanical systems, essentially a miniature machine that has both mechanical and electronic components on board. We utilize two different sizes, one French, which is 0.33 of a millimeter, and three French, which is 1 millimeter, for all of our catheter models. So the sensor then gets mounted into a case ready for wiring. Wires are attached to the sensor. They are essentially the size of a human hair, which takes a unique skill set to master. And then finally, an epoxy is used to seal the sensor in place so as not to expose it to liquid, which would damage the sensor and cause it to fail. This is an extremely important part of the process. This seal is directly related to drift and sensitivity, and it is important to get consistent adhesion and to eliminate bubbles. If the seal is too thick, the catheter would have reduced sensitivity, and we wouldn't see those beautiful signals that Keith is getting in his blood pressure. If the seal is too thin, the catheter would have increased sensitivity. So increased sensitivity, however, it would have a short lifespan in liquid, so maybe minutes to hours instead of longer term. So finding the right balance here is key, and this comes from experience. So all MEMS-based catheter technology use some form of sealant, which is unique to application and product. So coming on to drift. So one of the main areas of discussion with potential clients is around drift and how to minimize it. So drift occurs when electronics heat up and parts are subjected to liquids of varying temperatures. So this is not specific to Millar technology, but to all pressure transducers. They all have some form of drift through the warming up process, and to minimize this, we advise a pre-soaking period to stabilize the catheter and signal. So the figures shown on the slide here are some of the lowest drift specifications in MEMS technology on the market today. So these figures are from our single-use clinical devices, and that to achieve these results with reusable catheters, handling and cleaning are key, which is why Millar and ADI work together to provide our customers with robust training and support options specific to catheter care. You'll see on the middle cell, it shows drift levels of less than 1 mil of mercury over 7 days, with a maximum drift of 2.5 milligrams of mercury, which is directly due to the manufacturing processes described previously. Manufacturing and design play a huge role in order to consistently achieve these specifications. So these, as I mentioned, these are figures from our clinical production line, and specifications for each of our preclinical research catheters can be found in their instructions for use and are less than 6 mils of mercury over 12 hours. So the most widely asked question I get asked when I meet new clients are, in which animal models can Millar pressure sensor technology be utilized, and in which applications can I use them? So we often mention mouse to man, as this is appropriate when discussing the first question with researchers. So we have an extensive range of catheters starting from 1 French, which is 0.33 mils of mercury, millimeters in diameter, through to larger 5 French models, which are 1.6 mils in diameter. We provide single and dual pressure models, as well as catheters with straight, curved, and pigtail options, depending on access requirements. And if you have any questions around catheter selection, ADInstruments can help you with this. So now that we have established that catheters can be placed in most animal models, the question around applications is very similar. So Millar is widely recognized as a gold standard for preclinical cardiovascular pressure measurements. And when I'm asked the question around applications, this could be anything. So Millar pressure-only catheters measure an extremely accurate pressure wherever the researcher places it. So it is not an application-specific tool. So what I mean by this is, for example, a researcher could utilize a Millar catheter in left heart experiments, and then they can use the same catheter, dependent on size, to measure airways pressure if they wanted. They could also use the same catheter in a mouse or a rat or a larger animal if the size allowed it. So looking at the graph on the screen, some of the main areas of research utilizing our pressure catheter technology are listed there. And they include intracranial pressures, intraocular pressure, spinal, intradiscal pressures, just to mention a few. So now I wanted to move on to more of an applications insight, specific part of the talk. And so today we're just going to focus on intracranial pressure, IFP, airways pressure, with a small focus on sleep apnea. And we are limited by time, so I'm just going to give a brief background on the application and some ideas on how to place the catheter from publications. So I wanted to firstly focus on ICP. Why, may you ask? So ICP is the largest application by quantity of catheters supplied by Millar every year. It's much larger than cardiovascular and sleep, and it's not very widely known in the market. So our technology is extensively used in ICP applications through an OEM partnership with a medical device manufacturer. So essentially we're placing our sensor into their product, their catheter. So just to provide some background, in 1990, a medical device manufacturer approached Millar to develop a better method of measuring ICP. So fiber optics and fluid fill technology were not appropriate due to the bending requirements of the catheter on access to the site, due to being around 90 degrees. So Millar technology was selected to be integrated into the catheters due to it being insusceptible to motion and patient position, along with meeting bending needs of the device. Its low drift over days and accuracy at low pressure were also contributing factors. So we continue to supply ICP catheter technology to this day to traumatic brain injury patients across the globe. So there are a number of measurement challenges when recording ICP, the main one being accessing the most appropriate site. So in animal models, historically, acute measurements have been made over short periods of time. So we're talking hours here, where holes are drilled to access the dura and the Millar catheter are placed in situ. So ideally, measurements need to be taken over a period of days to weeks to regulate intracranial pressure and cerebral perfusion pressure, as it is fundamental in providing a stable environment to enable normal brain function. So one way that has been developed over the past 5 years to address the longer term recording needs is with wireless telemetry solutions. So since 2015, implantable devices fitted with Millar catheters, I think it's a 2 French model, have been utilised to record chronic measurements of ICP in rat models. The original study referenced on this slide was for a 28-day implantation, continuous recording in a freely moving rat by Dr. Sarah Jane Guild from the University of Auckland. So as you can see in the schematic, it is quite an involved surgery to place the catheter. Holes are drilled for screws, which are used for anchoring, and an additional access hole is needed to insert the catheter, in this case under the dura. Gel foam and cement is then used to secure the catheter in place. This is required to make sure the catheter does not move over the implanted period of time. So groups are currently doing ICP research in Europe with these wireless telemetry devices, and there have been some publications recently released. They are based around hypertensive response to ischemic stroke and blast-induced traumatic brain injury. One thing to note with all research to do with recovering animal models is that good sterile surgery is a key factor in success. Recovery of animals normally takes around 5 to 7 days, after which continuous long-term data can be collected. So the second application that we wanted to explore today is IFP, or tumor pressure. So elevated interstitial fluid pressures in human and rodent solid tumors have been recognized as an indicator of reduced therapy response and decreased free survival rates. So Millar technology has been validated by Ozodem and Hagens in their publication, A Simple Method for Measuring IFP in Cancer Tissues from 2005. So Millar's microtip catheters provide a simple, accurate measurement for IFP, and they offer a varying IFP response in a variety of models. So to date, Millar's SPR 1000, which is our 1 French model, SPR 320, which is our 2 French model, and SPR 524, high fidelity pressure catheters have all been used to deliver accurate, high quality IFP tumor measurements in publications. So how is tumor IFP measured? To begin with, it is much more straightforward than ICP. So tumor IFP can be measured easily by inserting a Millar pressure catheter directly into the tumor pressure. It involves inserting a calibrated pressure catheter into the tumor of the anesthetized animal. Often a guide needle is used to penetrate the tumor or tissue of interest first. Then the Millar pressure catheter is inserted over the needle. The needle is then retracted along the catheter cable. So ADInstruments has some great detailed videos. I think they're newly released on how to calibrate a Millar catheter, which can be found at ADInstruments.com. The following slides show some methods of insertion from publications. So this is the method of insertion from the validation publication from Hagens and Ozodem. It involves an SPR 320, which is a 2 French Millar catheter. It is inserted through an 18 gauge needle. The needle is introduced to the catheter, to the center of the tumor. The needle is then slowly retracted, leaving the Millar catheter in situ. The catheter can be marked for millimeter graduation using a standard fine point pen or a Sharpie along the length of the cable. Here was another publication from Mori showing a similar technique. Again, it shows a needle being placed into the tumor site. In this case, a 23 gauge needle for insertion of an SPR 1000, which is our 1 French catheter, smallest catheter by diameter. The guide is then removed and the catheter is left in situ. And finally, some more recent techniques have used peel-away needles. So this introducer provides the benefit of not having to retract the introducing needle back along the catheter cable. So this is a disposable single-use needle introducer. So the final application that we wanted to highlight today is airways pressures, which include a number of areas of study, such as those listed on the slide. So they include obstructive sleep apnea, central sleep apnea, non-apneic events, exercise-induced laryngeal obstruction, high-flow nasal O2 therapy, and vocal cord contact pressure. So Millar technology excels at measuring small pressure changes in low pressure environments. So as discussed previously, both ICP and IFP are low pressure applications, along with most airway applications where you would be looking at pressures in the region of, say, -10 to 30 mils of mercury. So all our sensors measure pressure changes by deformation strain. Some recent publications have placed the catheters in pleural spaces to measure pressures during high flow oxygen therapies. And we've also had success and validation recently of our technology for studying laryngeal pressures during exercise, as well as vocal cord contact pressures. So one of the most popular applications utilizing Millar pressure technology outside of cardiovascular is obstructive sleep apnea. So it's characterized by recurrent episodes of the cessation of breathing. The interruption in airflow is due to pharyngeal collapse caused by anatomical predispositions and insufficient neuromuscular compensation. So initial side effects of OSA include sleepiness and daytime fatigue. However, research has shown chronic OSA to cause high blood pressure, stroke, and even decreased life expectancy. Because this syndrome is a combination of anatomical and neuromuscular elements, predicting successful treatments is difficult. There are currently a number of clinical research studies ongoing utilizing Millar technology in these applications. And I've noticed due to COVID-19, respiratory research is developing quickly. So in saying this, I'm really looking forward to Dr. Fomenti's talk after this session. So placement. Placement of catheters for human applications, where in most cases the patient is awake, is done nasally through a bronchoscope with the aid of a decongestant and an anesthetic. For anesthetized animal models, the trachea is cannulated whilst the animal maintains spontaneous ventilation. Millar catheters are then inserted through the mouth into the esophagus and stomach, spanning the thoracic and abdominal surfaces of the diaphragm muscle, respectively. So correct catheter position can be determined based on the signal deflection and post-mortem analysis in an animal model. So when comparing Millar technology to traditional fluid-filled or, say, balloon pressure catheters, a number of differences are clearly seen. I've highlighted a few here on this slide, which include true pressure signal at source. So Millar catheters feature the latest sensor-based technology mounted at the distal tip where the pressure measurement is taken. So essentially, you're measuring pressure where the tip of the catheter is placed, as opposed to along a fluid-filled line that connects to an external pressure transducer. They're easy to position and keep in place for long periods of time, compared to balloon and fluid-filled pressure lines, which sometimes need maintenance and correction of inflation and removal of air bubbles. Their accuracy at low pressure, along with low drift, is a key. And they're also free from movement artifacts, so which sometimes can be seen with air bubbles in fluid-filled models. So this concludes my presentation. I just want to thank you for your time in attending the webinar today, and I hope the information provided will be helpful in your research going forward.